This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to an epic journey through Earth's deep past. Today, we explore the Great Oxidation Event, a billion-year struggle that turned a lethal poison into the fuel for complex life. We'll investigate the microscopic innovators that reshaped our atmosphere, the global catastrophes of fire and ice they triggered, and the tectonic forces that finally stabilized our world. From the Oxygen Holocaust to the boring billion, prepare to see our planet's history in a startling new light. Let's begin Chapter 1, The Hidden Cradles of Oxygen. Chapter 1, The Hidden Cradles of Oxygen. For the first half of our planet's existence, the Earth was a world that we would find utterly unrecognizable and immediately lethal. If you were to stand on a shoreline 4 billion years ago, you would not be looking out at a blue ocean under a blue sky. Instead, the atmosphere was a thick, hazy shroud of nitrogen, carbon dioxide, and methane. The oceans were rich in dissolved ferrous iron, giving the water a greenish tint, and the air lacked even a trace of the free oxygen that sustains almost all complex life today. This was the Archean eon, a time when the rules of chemistry and biology were fundamentally different. Yet, beneath this alien sky, a quiet revolution was beginning. We often think of the rise of oxygen as a sudden, cataclysmic event, a single moment where the lights were switched on and the world was transformed. However, recent scientific investigations into the deep past have revealed a much more complex and mysterious story. It is a story of localized pockets of life that were experimenting with the most dangerous waste product in history long before it ever reached the atmosphere. The traditional narrative of Earth's history places the Great Oxidation Event at roughly 2.45 billion years ago. This is the point where the geological record shows an unambiguous shift, where oxygen levels finally rose high enough to leave a permanent mark on the planet's chemistry. But for decades, a nagging question has haunted geochemists. How could such a massive change happen overnight? The answer, it turns out, is that it didn't. The seeds of this transformation were sown hundreds of millions of years earlier by a group of microscopic innovators known as cyanobacteria. These organisms developed what is arguably the most important biological invention in the history of the universe: oxygenic photosynthesis. By using sunlight to split water molecules, they gained access to an almost limitless supply of energy. But they also released molecular oxygen as a byproduct. Recent research has provided a much more specific time frame for this hidden evolution. Genomic analysis suggests that the first aerobic bacteria, those capable of using oxygen, emerged as early as 3.35 billion years ago. This represents a staggering 900 million year lead before the Great Oxidation Event actually took hold of the planet. This means that for nearly a billion years, oxygen-producing life and oxygen-consuming life were engaged in a microscopic tug-of-war in a world that remained, on the surface, completely anaerobic. If oxygen-producing life was present so early, why did it remain confined to the shadows for so long? One fascinating theory suggests that the early Earth's chemistry was actively stacked against cyanobacteria. In the Archean oceans, high concentrations of nickel and urea created an environment that favored methane-producing archaea. These methanogens were the titans of the early Earth, and their metabolic dominance gave them a decisive competitive advantage. They effectively suppressed the rise of cyanobacteria, keeping them relegated to marginal environments where the chemical conditions were just right for a different kind of survival. These marginal environments were the first oxygen oases, and scientists have recently identified a concrete, localized cradle for this early life: asteroid impact craters. When a massive asteroid strikes the Earth, it creates more than just a hole in the ground. It generates a long-lived hydrothermal system. Within these craters, such as the Hapchen crater, hydrothermal lakes provided a unique shielded environment. These lakes were rich in the minerals and energy needed for life, but they were also isolated from the broader hostile chemistry of the global ocean. Within these crater oases, cyanobacteria could thrive and produce oxygen in concentrated pockets. These were the laboratories of the Archean, where life could experiment with photosynthesis and develop the necessary defenses against its own toxic waste. To survive within these oases, early microbes had to evolve sophisticated defense mechanisms. Oxygen is highly reactive. It attacks proteins, lipids, and DNA. These early innovators developed enzymes like superoxidase dismutase and catalase to neutralize the harmful effects of oxygen, allowing them to persist in environments that would have been lethal to their anaerobic neighbors. This biological innovation was not happening in a vacuum. It was leaving a shielded record in the very foundations of the Earth. Breakthroughs in analyzing zircon crystals have revealed apatitite inclusions that provide a window into this deep past. These inclusions show a shift from reduced to oxidized magmas long before the Great Oxidation Event, suggesting that the surface environment was already beginning to influence the chemistry of the Earth's crust in localized areas. The evidence for these early whiffs of oxygen is written in the subtle chemical signatures of ancient shales. One of the most famous examples comes from the Mount Macrae Shale in Western Australia, which dates back to 2.5 billion years ago. When researchers analyzed these rocks using high-resolution chemostratigraphy, they found an enrichment of transition metals like molybdenum and rhenium. Under normal anoxic conditions, these metals are locked away in sulfide minerals in the Earth's crust. They only become soluble and wash into the ocean when they are exposed to oxidative weathering. The presence of these metals in 2.5 billion-year-old marine sediments is a smoking gun for the presence of free oxygen. It suggests that even before the atmosphere was oxygenated, there was enough O2 in the surface environment to start eating away at the continents. This early oxygen wasn't just confined to the very surface of the water either. Research into thallium isotopes has provided a window into what was happening much deeper in the water column. Thallium is sensitive to the burial of manganese oxides on the seafloor, a process that requires the presence of oxygen in the water. By looking at shales from the Transvaal supergroup in South Africa, which are roughly 2.65 billion years old, scientists have found evidence of widespread manganese burial. This suggests that oxygen oases weren't just thin films on the surface of rocks. They were substantial enough to oxygenate the water column over continental shelves, reaching down to the seafloor in some regions. This indicates that the production of oxygen was already a massive, regionally significant process, nearly 200 million years before the atmosphere finally turned the corner. Molecular biology further supports this early timeline. By mapping the tree of life, researchers have estimated that heme copper oxygen reductases, the essential enzymes that allow organisms to use oxygen, evolved during the Mesoarchean, between 3.2 and 2.8 billion years ago. This suggests that the ancestors of many modern bacteria were already oxygen-ready in a world that was still predominantly anaerobic. As soon as cyanobacteria created a little bit of oxygen in their localized oases, other microbes evolved the tools to interact with it. So why did it take so long for this oxygen to reach the sky? The reason lies in the sheer scale of the Earth's natural buffers. The Archean Earth was a world of sinks. The oceans were filled with dissolved ferrous iron, which acts like a giant sponge for oxygen. As soon as a cyanobacterium released a molecule of O2, it would react with the iron to form insoluble iron oxides, essentially rust, which would then sink to the bottom of the ocean. This process created the massive banded iron formations that characterize the geological record of this era. These formations are a literal record of the struggle between life's production of oxygen and the planet's ability to absorb it. In addition to the iron in the sea, the atmosphere was full of reducing gases like methane and hydrogen that would react with any oxygen that managed to escape the ocean. This chemical standoff meant that for nearly 1 billion years, oxygen was produced but immediately consumed. It was only when the methane greenhouse began to falter and the volcanic sinks began to change that the balance finally shifted. This new perspective changes how we look at the history of life. We are the descendants of a lineage that spent 1 billion years in these oxygen oases, refining the machinery of modern life in the shadows of a methane-heavy world. These early microbes developed the pigments to harvest light and the defenses to survive a corrosive gas, while the planet remained an alien, suffocating place. By the time the global transition finally occurred, the biological stage was already set. The enzymes were ready, and the metabolic pathways were established. The transition to an oxygenated world was not a sudden miracle, but a protracted struggle, a tug-of-war between the biological drive to produce and the geological forces that sought to consume. This struggle was deeply tied to the shifting of the Earth's very foundations and the movement of the continents themselves. Chapter 2: The Tectonic Tug-of-War. For a long time, the Great Oxidation Event was treated by science as a singular, sharp boundary in time, a moment when the Earth simply turned a corner and never looked back. We imagined it as a relatively quick transition, perhaps taking a few million years, where the atmosphere was flooded with oxygen and the modern world began. However, as our ability to peer into the chemical signatures of ancient rocks has improved, that simple picture has dissolved. We now know that the oxygenation of our planet was not a sudden event, but a grueling 200 million year struggle. It was a period of extreme instability, characterized by a geological and biological teeter-totter, where the atmosphere and oceans flipped back and forth between anoxic and oxic states in a series of violent fits and starts. To understand why this transition was so protracted and chaotic, we have to look far beneath the surface, into the very mantle of the Earth and the shifting of the tectonic plates. The evidence for this fits and starts transition is most clearly written in the sulfur isotopes found in marine shales from the Transvaal supergroup in South Africa and the Kola Peninsula in Russia. In an atmosphere without oxygen, ultraviolet radiation from the sun strikes volcanic sulfur dioxide, triggering a specific type of chemical reaction called mass-independent fractionation. This process leaves a unique isotopic fingerprint in the sulfur that settles into the sediments. For the first 2 billion years of Earth's history, this fingerprint is everywhere. When oxygen is present, even in small amounts, it forms an ozone layer that blocks the specific UV rays needed for this reaction, and the fingerprint disappears. In a simple one-way transition, we would expect to see this signature vanish once and then never return. Instead, the rock record shows something much more turbulent. The rare sulfur isotope signatures disappear, then reappear, then disappear again, multiple times over a period of roughly 200 million years, starting around 2.45 billion years ago. This tells us that the Earth wasn't ready to be oxygenated. Every time the cyanobacteria managed to push oxygen levels above a certain threshold, the planet's natural chemistry fought back. This phenomenon is known as bistability, the idea that the Earth's atmosphere has two stable states. One is a low oxygen, methane-rich state, and the other is a high oxygen state shielded by an ozone layer. During the Great Oxidation Event, the planet was caught in a violent jump between these two states, unable to find its footing. For nearly 200 million years, the oxygen levels fluctuated across a critical threshold of roughly 1/100,000th of the present atmospheric level. This was the tipping point where the chemical sinks were nearly saturated, but any slight change in geological activity could pull the oxygen right back out of the sky. One of the most significant factors in this struggle was the shifting nature of Earth's volcanism. During the Archean eon, the vast majority of volcanic activity happened underwater, at mid-ocean ridges and submarine hotspots. Submarine volcanoes are incredibly effective oxygen sinks. Because the lava erupts directly into the sea, the gases it releases, primarily hydrogen and hydrogen sulfide, interact with the water in a way that makes them highly reducing. They essentially act as a chemical sponge, soaking up every molecule of oxygen produced by life. However, as the Paleoproterozoic era progressed, the Earth's tectonic regime began to change. The assembly of a large continental mass, known as the supercontinent Kennerland, was underway. As these cratons collided and stitched themselves together, volcanic activity began to move from the deep sea onto the newly formed land. This shift to subaerial volcanism was a game-changer for the atmosphere. Gases released from land-based volcanoes are naturally more oxidized than those from submarine ones. When a volcano erupts on land, the gases are released at lower pressures and do not have the same immediate reducing interaction with seawater. As the ratio of subaerial to submarine volcanism increased, the overall sink for oxygen began to weaken. This tectonic transition wasn't smooth. It happened in pulses as continents drifted and merged, which contributed to the flickering, unstable nature of the oxygen levels. The specific equatorial positioning of the Kopfall Craton during this era further influenced how these new landmasses weathered and interacted with the global climate system. But the tectonic influence went even deeper, reaching into the Earth's mantle. Recent research has uncovered a fascinating lag between the interior of our planet and its surface. While life was producing oxygen at the surface, the mantle below remained highly reduced for billions of years. However, there is evidence of a mantle redox shift that occurred leading up to the Great Oxidation event. The smoking gun for this shift was found in the study of apatite inclusions trapped within ancient zircon crystals. These inclusions acted as shielded time capsules, protected from later chemical changes for billions of years. They show a clear transition from reduced to oxidized sulfur, proving that the chemistry of the Earth's interior was fundamentally changing. This theory suggests that a deep primordial layer of oxidized material had been trapped at the base of the mantle since the Earth's formation. This layer was likely created during the magma ocean stage, when the high pressures of the deep Earth caused iron to undergo a process called disproportionation, where it splits into metallic iron that sinks to the core and oxidized ferric iron that stays in the silicate mantle. This oxidized material was enriched with a mineral called bridgemanite, which is incredibly viscous. Essentially, it's very stiff and resists mixing. Because of this high viscosity, the oxidized layer at the bottom of the mantle was sluggish, taking nearly 2 billion years to slowly churn and mix upward into the upper mantle. It wasn't until around the time of the Great Oxidation event that this oxidized material finally reached the zones where magmas are generated. When it did, it changed the chemistry of the molten rock, causing volcanoes to tap into a source that was naturally more oxidized. While the mantle provided the long-term push, a secondary mechanism was irreversibly oxidizing the planet from the top down. Hydrogen escaped to space. This process was driven by the photolysis of methane. As ultraviolet light broke down methane molecules in the upper atmosphere, the resulting hydrogen, being light and unreactive, escaped the Earth's gravity and was lost to the cosmos forever. Because hydrogen is a powerful reducing agent, its permanent loss meant the Earth was losing its ability to neutralize oxygen. This provided a constant background oxidation that slowly saturated the planet's chemical sinks, eventually allowing the atmosphere to reach the bistability tipping point. The timing of these tectonic shifts is also tied to the appearance of large igneous provinces, or lips. These are massive, continent-scale volcanic events that can last for millions of years. One such event, the Ungaluk Large Igneous Province in southern Africa, has been dated to approximately 2.426 billion years ago. These lips played a dual role in the oxygen story. On one hand, they released massive amounts of nutrients like phosphorus into the oceans through the weathering of their basaltic rocks, fertilizing the seas and leading to surges in oxygen production. On the other hand, the sheer volume of volcanic gases they released could temporarily overwhelm the atmosphere, causing oxygen levels to crash back down. These chemical fluctuations triggered a series of global glaciations known as snowball Earth events, which further complicated the oxygenation process by shutting down the very biological productivity that was driving the change. See Chapter 4. This struggle is also reflected in the thallium isotope records of the Transvaal supergroup, which track the burial of manganese oxides as a proxy for early oxygenation around 2.65 billion years ago. The final stabilization of this system began around 2.22 billion years ago, marked by the onset of the Lomagundi carbon isotope excursion. This event represents a period of unprecedented organic carbon burial, where the reduced half of the photosynthesis equation was finally tucked away in the Earth's crust on a massive scale. However, the era of instability did not end overnight. The transition continued until the end of the Lomagundi event, roughly 2.06 billion years ago. By this point, the mantle had mixed enough oxidized material upward, the continents had grown large enough to provide massive burial grounds for carbon, and the subaerial volcanoes were no longer exhaling enough sinks to keep the oxygen down. As the atmosphere finally stabilized in its new high oxygen state, the life that had triggered this transformation was about to face the consequences of its own success, as the very gas filling the sky became a lethal toxin that would rewrite the tree of life. Chapter 3: The Oxygen Holocaust. We are accustomed to thinking of oxygen as the very essence of life, the invisible fuel that powers every breath we take and every thought we have. In our modern world, its absence is the definition of death. But if we could transport ourselves back to the middle of the Archean eon, we would find a biosphere for which oxygen was not a gift, but a terrifying and corrosive poison. To the billions of microbes that had ruled the planet for over 1 billion years, the rise of molecular oxygen was an industrial-scale pollution event, a biological disaster so profound that many scientists refer to it as the Oxygen Holocaust. This was not a gentle transition. It was a global chemical war that resulted in what was likely the most lethal mass extinction in the history of our planet, fundamentally erasing the dominant life forms of the early Earth and forcing the survivors into the dark, hidden corners of the world. To understand the scale of this catastrophe, we have to visualize the Earth as it existed before the cyanobacteria took over. This was a world dominated by anaerobic organisms, methanogenic archaea, and sulfate-reducing bacteria that thrived in an environment completely devoid of free oxygen. These organisms were perfectly adapted to a world of low energy and high stability, a world where the chemistry of life was based on the slow, careful manipulation of hydrogen, sulfur, and iron. There is even a compelling scientific proposal known as the Purple Earth hypothesis, which suggests that before the rise of green, chlorophyll-based life, the planet may have been dominated by microbes that used a molecule called retinol to harvest sunlight. Retinol absorbs green light and reflects red and violet, which would have given the shallow seas and microbial mats of the Archean a distinct purple hue. The arrival of oxygen-producing cyanobacteria shattered this stability. Cyanobacteria were the ultimate biological disruptors. By evolving the ability to perform oxygenic photosynthesis, they tapped into the most abundant electron donor on the planet, water. This gave them an almost infinite supply of fuel, allowing their populations to explode across the globe. But the byproduct of this process, molecular oxygen, was a chemical weapon. Oxygen is one of the most reactive elements in the universe. It is a powerful oxidizer, meaning it has a violent tendency to steal electrons from other molecules. In a world that had never known free oxygen, the organic compounds that made up the bodies of living cells, their proteins, their lipids, and their very genetic material were completely defenseless against this chemical attack. The transition was not a steady climb, but a precarious teeter-totter between oxygen production and destruction. For millions of years, the planet's natural chemical sinks, the dissolved iron in the oceans and the volcanic gases in the sky, struggled to soak up the new gas. This created a period of extreme instability where oxygen levels flickered in fits and starts. When oxygen managed to gain a temporary foothold, it reacted with transition metals like iron, which were abundant inside ancient microbes, to create reactive oxygen species. These include superoxide radicals, hydrogen peroxide, and the dreaded hydroxyl radical. These molecules were the biological equivalent of shrapnel. They did not just cause general decay, they specifically targeted and disabled the iron-sulfur clusters that sit at the heart of anaerobic metabolic enzymes. To an anaerobic microbe of the Archean, a whiff of oxygen was like a spray of acid, rusting the machinery of life from the inside out and tearing through cellular membranes. The cyanobacteria themselves were not immune to this danger. In the early stages of their evolution, the oxygen they produced was just as toxic to them as it was to their neighbors. This created a paradoxical crisis for the producers. Modern cyanobacterial mats show that these organisms often consume nearly as much oxygen at night as they produce during the day, a desperate metabolic dance to keep the concentrations of the gas from reaching lethal levels within their own colonies. The Great Oxidation Event was, in a very real sense, a case of a species poisoning its own environment so thoroughly that it nearly wiped itself out along with everything else. Quantifying the scale of the resulting extinction is one of the greatest challenges in paleontology. Because the victims were all microscopic and lived billions of years ago, they left no skeletons or shells for us to count. However, the geochemical record provides chilling clues. Isotope geochemistry data from sulfate minerals indicate a decrease in the total size of the Earth's biosphere by more than 80% at the end of the Great Oxidation Event. This suggests a collapse of life on a scale that dwarfs the more famous extinctions of the later Phanerozoic eon. The thriving, diverse world of the Archean was systematically dismantled. The methanogenic archaea, which had likely been the primary regulators of the Earth's climate, saw their habitats shrink and their populations crash. The retinal-using organisms of the Purple Earth were largely outcompeted and pushed toward the brink as the green cyanobacteria monopolized the sunlight and filled the water with poison. The survivors of this Holocaust were forced to find refugia, places where the reach of oxygen could not penetrate. We can still find their direct evolutionary heirs today, living fossils of a lost world. They thrive in the deep, black mud of stagnant ponds, in the waterlogged soils of marshes, and around the boiling hydrothermal vents of the deep ocean floor. They even found a new, strange home in the digestive tracts of the animals that would evolve hundreds of millions of years later. These anaerobic lineages occupy the niches where the planet's ancient, reduced chemistry still holds sway, a reminder that for the first half of Earth's history, our current atmosphere would have been considered a death sentence. But the Oxygen Holocaust also triggered some of the most brilliant and transformative adaptations in the history of life. As the world became increasingly oxygenated, a few lineages of bacteria didn't just learn to tolerate the poison, they learned to harness it. They evolved a suite of protective enzymes, such as superoxide dismutase and catalase, to neutralize reactive oxygen species. Some survivors even developed proteins to bind oxygen into melanin, using the pigment as a shield to protect their internal structures from oxidative damage. These ancient oxygen-binding proteins, such as tyrosinase, would eventually evolve into respiratory pigments like hemocyanin, the blue blood found in many modern invertebrates. The most significant adaptation, however, was a new form of metabolism called aerobic respiration. This was a metabolic masterstroke. By using oxygen as a terminal electron acceptor, organisms could extract far more energy from their food than was possible through anaerobic fermentation. While an anaerobic microbe might get only 2 molecules of ATP, the cell's energy currency, from a single molecule of glucose, an aerobic microbe could get as many as 36. This 18-fold increase in power was the energy bribe that allowed life to move past a simple, single-celled existence. This adaptation led to the most important merger in the history of complex life, symbiosis. During the height of the oxygen crisis, an anaerobic archaeal host cell engulfed a smaller, aerobically respiring proteobacterium. Instead of digesting the smaller cell, the host kept it alive in a permanent symbiotic relationship. The smaller cell provided the host with a massive surge of energy and a way to detoxify the surrounding oxygen, while the host provided protection and nutrients. This engulfed bacterium eventually evolved into the mitochondrion, the powerhouse of the cell found in every plant, animal, and fungus on Earth today. Our very existence as complex, multicellular beings is a direct result of this ancient deal struck to turn a lethal poison into a source of unprecedented power. The Great Oxidation Event also began to shield the surface of the planet. As oxygen accumulated, it enabled the formation of a thin ozone layer, providing a vital defense against ultraviolet radiation that, combined with the presence of oxygen, would have otherwise been doubly lethal to early life. This shield, along with the newfound metabolic wealth of aerobic respiration, set the stage for life to eventually leave its deep-water refuges. However, the chemical victory of oxygen over the ancient atmosphere came with a devastating environmental cost. As the new gas scrubbed the methane from the sky, the planet's primary heat-trapping mechanism began to vanish, threatening to turn the Oxygen Holocaust into a permanent global deep freeze. Chapter 4: The Methane Collapse and the White Sky. While the biological and chemical warfare of the Great Oxidation Event was unfolding in the oceans, a second, perhaps even more dramatic crisis was gathering in the sky. Under a faint young sun, a star shining with only 70% of its modern intensity, the Earth's climate was precariously dependent on a thick blanket of greenhouse gases. As established in the earliest days of the Archean, the most vital component of this insulating shroud was methane. Produced in massive quantities by methanogenic archaea, this methane acted as a planetary thermostat, keeping the surface temperature well above freezing despite the sun's low output. It created a hazy, orange-tinted world, but it was a thermostat with a fatal vulnerability to oxygen. As cyanobacteria began to flood the world with free oxygen, they were inadvertently dismantling the planet's heating system. The rise of oxygen did not just poison the anaerobic microbes of the sea, it systematically scrubbed the methane from the atmosphere. This chemical transition was essentially like swapping a heavy winter parka for a thin linen shirt in the middle of a blizzard. The consequences were immediate and catastrophic, triggering a period of cooling so severe and so long-lasting that it remains the most significant climatic event in the history of the planet. The first tremors of this cooling were felt during the Pongola glaciation, the earliest major ice age in the geological record, which served as a chilling precursor to the much longer Huronian event. Beginning around 2.45 billion years ago, the Earth entered a deep freeze that would last for nearly 300 million years. This was the Huronian glaciation, a series of snowball Earth events where ice sheets did not merely advance from the poles, but potentially encased the entire planet in a shell of white. The evidence for this global freeze is written in the rocks of the Huronian supergroup in Canada and the Transvaal supergroup in South Africa. Geologists have identified layers of glaciomarine diamictites, rocks formed from the debris dropped by melting icebergs in regions that were, at the time, located near the equator. One specific formation, the Makjanyane diamictite in South Africa, is the smoking gun for the snowball Earth hypothesis. Paleomagnetic data indicates that these glacial deposits were laid down at a latitude of only 11 degrees. For ice to exist at sea level so close to the equator, the entire planet must have been in the grip of a truly global winter. The onset of this freeze was accelerated by a powerful phenomenon known as the ice albedo feedback loop. Ice is highly reflective. It has a high albedo, meaning it bounces most of the sun's energy back into space. As the methane greenhouse collapsed and temperatures dropped, ice began to form at the poles and on high-altitude mountains. This white ice reflected more sunlight, which cooled the planet further, allowing more ice to form. Once the ice sheets reached a critical threshold, roughly 30 degrees latitude, the feedback loop became unstoppable. The planet rapidly spiraled into a total freeze, with temperatures at the equator potentially dropping to -50 degrees Celsius. Recent research suggests that the timing of this collapse was also influenced by a nickel famine. Early methanogens relied on nickel as a vital cofactor for the enzymes they used to produce methane. As the Earth's interior cooled and the style of volcanism changed, the supply of volcanic nickel began to dwindle. Between 2.7 and 2.4 billion years ago, nickel concentrations in the ocean dropped by a factor of 400, starving the methane producers. However, this famine was somewhat buffered by an uptick in sulfide weathering at the start of the Great Oxidation event. This provided just enough of a chemical cushion to prevent the Earth from falling into an even more catastrophic and permanent icehouse state, allowing the planet to eventually find a way back from the brink. The struggle for stability was further complicated by the feedback of atmospheric bistability and the emergence of an ozone layer, which created a climatic teeter-totter, where the planet's temperature and chemistry flickered between two extremes for millions of years. The tectonic configuration of the Earth, including the assembly and equatorial positioning of the supercontinent Kenorland, also played a crucial role in pushing the planet into the ice by increasing the rate of silicate weathering and reflecting more solar energy back into space. The struggle for life during this 300-million-year winter was immense. With the oceans covered in ice, perhaps kilometers thick in some places, the productivity of the cyanobacteria was severely limited. They were likely restricted to small polynyas, areas of open water kept liquid by volcanic heat, or to the thin layers of meltwater on top of the ice. The entire biosphere was forced into a survivalist mode, clinging to life in hydrothermal vents or in the narrow cracks of the frozen seas. This period of extreme cold acted as a secondary filter, following the initial oxygen crisis. Life had to survive not only a corrosive atmosphere, but also a world that had become a desert of ice. However, the Earth's internal heat provided the eventual escape route. Even while the surface was frozen, volcanoes continued to erupt beneath the ice and on the few exposed patches of land, exhaling carbon dioxide into the atmosphere. In a normal world, this carbon dioxide would be removed by rain and rock weathering. But in a snowball Earth, the hydrological cycle is essentially frozen. There is no rain to wash the CO2 out of the sky. Over millions of years, the concentration of volcanic carbon dioxide built up to staggering levels, perhaps hundreds of times higher than modern concentrations. The Ongeluk Large Igneous Province in South Africa provides a specific window into this transition, where the long-term buildup of volcanic carbon dioxide helped drive the planet toward the eventual thaw. Eventually, the greenhouse effect from this massive CO2 buildup became strong enough to overcome the reflectivity of the ice. The thaw would have been just as violent and rapid as the freeze. As the ice began to melt, the dark oceans were exposed, absorbing heat and accelerating the warming. The Earth would have swung from a global refrigerator to a global sauna in a matter of centuries or millennia. This transition is recorded in the geological record by cap carbonates, thick layers of limestone that were deposited immediately on top of the glacial debris. These carbonates represent the massive washout of all that accumulated atmospheric CO2 as the rains finally returned and weathered the newly exposed continents. This cycle of freezing and thawing happened at least 3 and possibly 4 times during the Huronian period. Each time, the atmosphere and oceans were pushed into a state of extreme chemical and climatic flux. The banded iron formations also serve as a permanent geological legacy of this era, acting as a metronome of the ancient world that marks the oscillations of a planet struggling to find a new equilibrium. The final exit from the Huronian glaciations occurred around 2.22 billion years ago, coinciding with the onset of the Loma-Gundi carbon isotope excursion. While the final glaciers retreated at this point, the broader chemical transition of the Great Oxidation Event would continue for another 160 million years before the planet reached a new oxygenated equilibrium around 2.06 billion years ago. As the ice finally retreated for the last time, the world that emerged was fundamentally different from the one that had entered the freeze 300 million years earlier. The methane greenhouse was gone for good, replaced by a more stable, though less potent, carbon dioxide and oxygen-based system. The boring, gray world of the Archean had been replaced by a more vibrant, oxidized world, but one that was now much more vulnerable to the whims of the sun's luminosity and the Earth's own internal cycles. The stage was now set for a long interval of relative environmental stability, but this stability would bring its own set of challenges for the life that had survived the fire and the ice. How would the biosphere respond to 1 billion years of chemical stasis, and what hidden refinements were taking place beneath the boring billion? The stabilization of oxygen in the atmosphere following the chaos of the Great Oxidation Event did more than just change the color of the sky. It fundamentally rewrote the energy budget of the entire planet. While the transition had been a period of mass extinction and climatic upheaval, the world that emerged on the other side possessed a chemical potential that had never existed before. For the first 2 billion years of Earth's history, life had been limited by the relatively low energy yields of anaerobic metabolism. Fermentation and other oxygen-free processes are survival strategies, but they are not particularly efficient. In the presence of oxygen, however, a new metabolic pathway became possible: aerobic respiration. This was the true legacy of the Great Oxidation Event. Aerobic respiration allows an organism to extract an order of magnitude more energy from a single molecule of glucose than is possible through anaerobic fermentation. This massive energy surplus acted as a biological venture capital fund, providing the metabolic wealth necessary for life to invest in complexity. We see the first major returns on this investment approximately 2.3 billion years ago, when cyanobacteria began evolving into multicellular organisms, a transition enabled by the rise of atmospheric oxygen. This leap in organization allowed for specialized cell functions and the first tentative steps toward the macroscopic world. This energy revolution was driven not only by biological innovation, but by the physical evolution of the planet itself. During the Earth's formation, the planet spun much faster, with a day lasting only about 6 hours. By the time of the Great Oxidation Event, the Moon's tidal pull had slowed Earth's rotation to approximately 21 hours. This lengthening of the photoperiod was a critical physical driver of oxygenation. Longer days allowed cyanobacteria more uninterrupted sunlight to produce an oxygen surplus. In the shorter days of the Archean, the oxygen produced during the brief light hours was often consumed by the microbes' own respiration during the long nights. As the days stretched out, the oxygen production began to outpace consumption, allowing the gas to diffuse into the water column and eventually the atmosphere. This physical slowing of the Earth acted as a throttle, gradually opening to allow the biological engine to breathe and the oxygen to finally escape the confines of the microbial mats. The immediate world created by this surge of oxygen would have been unrecognizable and harsh. As oxygen began to attack surface minerals like pyrite, the planet likely resembled a global version of the modern Rio Tinto in Spain. This acid rock drainage would have been the norm across the continents. The landscape was characterized by extremely low pH levels, a distinct red tint from oxidized iron, and the massive release of heavy metals into the water systems. It was a world of corrosive runoff and toxic runoff, where the first inhabitants of the land had to contend with a chemical environment that was as aggressive as it was new. This period of intense oxidative weathering was the price the planet paid for its new atmosphere, a global-scale chemical reaction that turned the continents into sources of both essential nutrients and lethal toxins. This world of corrosive runoff was the crucible in which the survivors of the Great Oxidation Event had to forge their future. While the energy advantage of oxygen eventually drove the rise of the eukaryotic cell, allowing life to pay the metabolic tax required for larger genomes and complex internal structures, the planet did not immediately transition into a high-oxygen paradise. Instead, following the end of the Lomagundi carbon excursion around 2.06 billion years ago, the Earth entered a transitional phase known as Stage 3, beginning around 1.85 billion years ago, which led into the Boring Billion. Lasting from roughly 1.8 billion to 800 million years ago, the Boring Billion is characterized by a lack of major fluctuations in the carbon isotope record and a seemingly slow pace of evolutionary change. While previous estimates suggested oxygen levels were between 1 and 10% of present atmospheric levels, more recent geochemical proxies suggest they may have been as low as 0.1%. This extremely low concentration explains why complex life appeared to stall for 1 billion years. The deep oceans remained largely anoxic and rich in dissolved iron, a state known as ferruginous, while the surface waters held only a thin ribbon of oxygen. This stratified ocean structure limited the availability of key nutrients like molybdenum and phosphorus, keeping the biosphere in a state of low productivity stasis where life was essentially waiting for the next geological trigger. The end of this long stasis was marked by a second major rise in oxygen known as the Neoproterozoic Oxygenation Event, NOE, which began around 850 million years ago. We can track this second surge through specific geochemical proxies that distinguish it from the earlier Great Oxygenation Event. Strontium isotopes in the rock record indicate a massive increase in continental weathering, likely driven by the breakup of the supercontinent Rodinia, which washed essential nutrients like phosphorus into the oceans. Simultaneously, chromium isotopes provide evidence of manganese dioxide catalysts, which only form in highly oxygenated environments. This second event brought oxygen levels up to near-modern concentrations and was the final prerequisite for the evolution of large, complex animals. It was the moment the energy ceiling was finally lifted, allowing life to explore the possibilities of size and movement. The link between oxygen and the size of organisms is fundamental. Because oxygen must diffuse through tissues, the amount of available oxygen sets a hard limit on how large and thick an organism can be. In the low-oxygen world of the Boring Billion, life was mostly restricted to thin films and microscopic structures. But as oxygen levels surged during the Neoproterozoic, life was suddenly able to grow. The subsequent Cambrian explosion, which saw the sudden appearance of almost all major animal groups, was the ultimate realization of the energy potential first unlocked 2 billion years earlier. Today, we live in a world where oxygen is so ubiquitous that we treat it as a fundamental property of the planet. But the geological record reminds us that the oxygenated atmosphere is not an equilibrium state. It is a continuous industrial output of the biosphere. If photosynthesis were to stop tomorrow, the free oxygen in our air would be scrubbed out by reactions with rocks and volcanic gases in just a few million years. Our breathable air is a steady-state byproduct of a microbial process that has been running for billions of years.
However, scientific models of the Earth's far future suggest that this oxygenated era is only a temporary chapter in the planet's history. As the Sun continues to age, it is slowly getting brighter and hotter, a consequence of the increasing density of its core. In about 1 billion years, the Sun's increased luminosity will cause the Earth's surface temperature to rise, accelerating the weathering of silicate rocks. This process is a powerful carbon sink. As rocks break down, they react with carbon dioxide, pulling it out of the atmosphere to be buried as carbonates on the ocean floor. Eventually, carbon dioxide levels will drop so low, below the threshold of approximately 10 parts per million, that plants will no longer be able to perform photosynthesis.
When the plants die, the primary source of oxygen will vanish. Simulations of this future Earth indicate that the atmosphere will undergo a catastrophic deoxygenation event, returning the planet to a state very similar to the Archean eon. Oxygen levels will crash by a factor of over 1 million, and the methane-producing microbes that have been hiding in the shadows for billions of years will once again become the dominant life forms. This return to an anoxic, hazy state will be the final mass extinction for complex life, as animals find themselves in an environment that has become as toxic to them as the early Earth was to their anaerobic ancestors. The Great Oxidation Event will have been a long, brilliant surge of energy in the middle of a much longer history of anoxia. We are living in a rare and beautiful window of time, a high-energy era enabled by a microbial accident that turned a poison into a fuel. The study of this event offers a sobering perspective on the relationship between a species and its environment. The cyanobacteria did not intend to change the world. They were simply successful at harvesting energy. Yet their success led to a global reorganization of the planet's chemistry. The presence of oxygen in our atmosphere today is a testament to the resilience and innovation of life. It is the residue of a battle that was fought across every ocean and every continent. We carry the legacy of this event in every cell of our bodies and in the iron-rich blood that carries it through our veins. The Great Oxidation Event was the moment the Earth grew up, a violent and transformative coming of age that set the stage for everything we know. The inevitable future return to an anoxic state serves as a reminder of the transience of our current environment. The oxygenated world we inhabit is a temporary feature of a dynamic planet, a high-energy interlude in a much longer history of chemical reduction. Our existence is defined by this temporary energy surplus, a gift from the microscopic polluters of the deep past. The same chemical reactivity that once nearly wiped out life is now the very thing that makes our complex, high-energy existence possible. Even as the Sun's aging process eventually pulls the oxygen back out of the sky, the record of what life achieved during this era will remain written in the minerals and isotopes of the Earth's crust. If the history of our planet is a book, we are currently reading the most vibrant, high-action chapter, but the final pages have already been plotted by the same laws of chemistry that wrote the beginning. What will remain of the biological legacy when the lights of the oxygenated world finally dim? The Great Oxidation Event was not a sudden transformation, but a billion-year struggle between microscopic innovators and the geological forces of a young Earth. This Oxygen Holocaust dismantled an ancient world, yet provided the metabolic wealth required for complex life to emerge. We are the products of a biological revolution that turned a lethal poison into the very fuel for our existence. However, the geological record reveals that our oxygenated atmosphere is a temporary industrial output of the biosphere, destined to eventually fade as the planet returns to its anaerobic origins. We live within a rare, high-energy window, a vibrant chapter in a much longer history of chemical reduction. As we look to the stars for life, we must remember that our breathable air is a hard-won, fleeting gift from the deep past. Thank you for joining us on this journey through Earth's transformative history.